We are the CEMIOS team from the Lucerne University of Applied Sciences and Arts. Within the REXUS program, we propose to fly frog eggs on board of a sounding rocket in order to study the properties of mechanosensitive ion channels under microgravity conditions. Our research will help to understand how cells can sense forces.
The core idea is that we want to investigate why our body, especially tissues, that they're particularly sensitive to mechanical stimulation, why and how they are responding to this mechanical force that is working, and then, for example, start growing. An example is when we go to a gym, when we do our trainings there, our muscles start to grow. So this is well known and of course quite often used, but we're not sure what's really going on cellular level. So why the cells and how the cells are responding to the mechanical forces. Well, the idea that going to space is we actually remove all the physical forces on the membrane to study the system on an unbiased and unloaded situation, and that gives us very good information for then going further to actually investigate then the situation under a situation where we have forces working.
We are interested in studying the property of mechanosensitive ion channels under microgravity condition. In order to do this, we need a cell which expresses the mechanosensitive ion channel. We use a frog egg, which we refer to as oocyte, to do this. The reason for it is the oocyte is fairly large, it's approximately 1 millimeter in diameter, and it's very robust. The essential measurement was done in a device like this, which was developed by Daniel Schaffhauser. The oocyte is fixed in a funnel with a tiny hole at the bottom. This isolates a patch of the cell membrane. Now we are interested in determining the current voltage relationship across that patch. We have 4 electrodes to do this. The 2 electrodes at the top allow us to control the voltage. The 2 electrodes at the bottom allow us to determine the current. So we can determine the current voltage relationship. The signal we get from such a reading is not the signal just from the mechanosensitive ion channel, but also from a background. So we need to determine the background signal. In order to do this, we can exchange the liquid in the lower compartment. This allows us to apply a drug which will specifically block the mechanosensitive ion channel. So we do 2 readings, one without the blocker and one with the blocker. The difference of the 2 readings will give us a signal from the mechanosensitive ion channel.
The voltage which shall be applied to the frog egg is set by the board computer software and is then sent to the measurement unit, and that voltage is applied to the frog egg by a electronic circuit which exactly holds the voltage on that value, and then the current through the membrane is measured, is digitized and then processed by the software.
Hello together. I'm going to talk about verification. So we only have one start, so we have to make sure that everything really goes well. And to do so, we'll do a lot of testing, but also computer simulations, of course, and lots of calculations. So the first thing, or maybe the biggest problems, is vibrations. So everything you want to launch with the rocket, you always have the problem with the vibrations. So you have to make sure everything is really fixed really tight, and you also have to make sure that your structure is not falling apart if it's really shaken. So we'll definitely have to do a test on the shaker table, but we also do simulations to make sure everything goes well. Then a second point, of course, is acceleration. The rocket will accelerate at 20 Gs, which is really a lot. And yes, the structure has to be strong enough to withstand those forces. And the third point is temperature. So it's a biological experiment. We have to make sure that the temperature is around 20 degrees Celsius.
Hello, I will talk about the mechanical build-up of our experiment. This is a wooden mock-up of our experiment. It consists of 3 levels. On the bottom floor there is the fluidic system with the gas cartridge that will be mounted on the side, pressure controllers and the other stuff. Then in the center section there are our 2 late access modules where the oocytes are placed. The oocytes are in 3 chambers placed in the late access module, and they can only survive for about 5 hours in the chambers. So they have to be inserted shortly before launch, and that will happen approximately 1.5 hours before launch. Through a late access hatch they will be inserted into our module, and on top there is the electronics which will control the whole experiment. This is our schematics of our experiment. On one side you can see we have a pressure bottle, servo controllers and liquid containers. Those are on the bottom side of our experiment. The pressurized air is used to drive the liquid. The pressure regulators apply a certain pressure on the liquid. The liquid is controlled with valves and is applied to the oocyte. And there's a second pressure regulator which is used for the top pressure to hold the oocyte down into its place. Everything is controlled with the microcontroller, and all the used liquid is stored in the waste bag.
The board computer controls the whole experiment and communicates with the measurement unit to collect the measurement data. These data are stored to the 2 SD cards, which are recovered after the flight. Further, there are 2 acceleration sensors to correlate the measurement data to the microgravity phase. At last, there is a communication with the rocket service module, so we send data down to our ground station.